Hello, everyone. And thank you for your interest in the Carter Foundation and the kinds of research that we're conducting with regard to hereditary spastic paraplegia. I'm Joe Avalone, the president of the foundation, and I'm with me today is Dr. Craig Blackstone, who is our senior scientific advisor. He's a professor of neurology at Harvard Medical School and has been involved for many years in research into hereditary spastic paraplegia. We're very pleased to have him advising us throughout in our research programs. I wanted to spend a minute or two to just talk about what the mission of the Carter Foundation is, and then really we'll move on and have Craig explain to us where our various projects are and how things are going. So if you could go to the mission statement, please. Thank you. As you may know, the primary mission of the Carter Foundation is to investigate potential therapies for hereditary spastic paraplegia. And we have been focused on SPG3A and SPG4, which together make up well over half of the known patients who currently have hereditary spastic paraplegia. We've also been trying to understand the differences between early onset of this genetic disease and other conditions which start later. As there many years of research have gone into the basic science understanding of these diseases at the cellular level. A lot of it was done at NIH, and much of it was done under the direction of Dr. Blackstone and other centers around the country and indeed around the world. But what we are trying to do is to go the next step, which is to take that basic understanding at the cellular level and try and advance it into animal models, and in particular, ultimately a mouse model that can show the defects of the mutations and then show how treatment might improve it. That is called translational medicine, and that's the important part, the important next step that we're trying to undertake in order to move towards the actual treatments of the children that have this disease. If we end up having a reproducible model that can show the defect and then also show the effective treatments, it could be very attractive, in fact, necessary for either drug companies or institutes or others to take it up and then try and move therapies along. So this translational medical gap is what we're trying to solve with our research projects. If we could go to the next step, the next slide, please. The first set of projects done by Dr. Jun Lee are called iPSC projects, which it really refers to pluripotential stem cells. The stem cells that you've probably heard of have been able to be promoted into human neurons with the exact mutation of SPG3A. And we've been running some experiments looking to understand potential drug treatment options. I wonder if I could ask Dr. Blackstone to comment on the status of this research now.
Sure, Joe. Thanks for having me again. So there's, as you have listed here, there's a couple main projects, and as you outlined, one of our goals is to create new models as part of the translational studies and to be able to test new therapies. One advantage of iPSC models, even though they're cells in a dish, is that they're human cells in a dish, and that they're based on either cells from a patient who has this disorder, or we've engineered in the same type of mutation into a human line. And that allows us to see how human neurons are impacted by different therapies. And as you have outlined here, we have from our basic science studies several pathways that we think are promising in terms of therapeutics, and one of them is targeting lipid metabolism, and that's a key one. And then as you have here under project 2, it is sort of the same. It's a longer kind of a process, not just creating the model, but actually testing various types of therapies. And these are not just going to be linked to lipids. There's a few other pathways as well, including the BMP or bone morphogenetic pathway. So there's several of these pathways, and they're very important because we have abnormalities that we see in these human cells in a dish. the mutation causes abnormalities, and we can see whether these therapies that we're testing can reverse those abnormalities. That doesn't necessarily mean they'll work in people, but it's an important first step. If we see an abnormality and can treat that and restore that in a culture dish, that gives us the confidence to move on to other animal models, which of course take longer, cost more money. So these sort of experiments are critical to really sort of honing in on the best pathway to pursue for the larger animal-based studies.
Thank you, Craig. If we can move on to the next slide, please. The next set of experiments are being done in Drosophila, which is the scientific name for fruit flies. Dr. Nemo Sullivan at the University College Dublin, and once again, a colleague and under the direction of Dr. Blackstone, has been using this basic model to try and understand some key aspects of the disease. You might say, why fruit flies? And really, it has a long history in genetic discovery and scientific endeavors. But the two big advantages of using the fruit flies for these experiments for us is that they replicate fairly rapidly, like every 3 weeks. So you can have generations of genetic mutations, et cetera, within a relatively short period of time. And they happen to have along their wing a very long neuron that at some level acts like a human upper motor neuron, which is the neuron that has the biggest problems in the SPG3A, SPG4, and in fact, all the hereditary spastic paraplegia. So as unlikely as it seems that we could learn much from a fruit fly, we can learn very much from the fruit flies. What we've been looking here is to try and understand the differences between the de novo onset, that is the neonatal onset that comes from de novo without either parent having the genetic defect, versus the other, even more common, of which one of the parents is carrying the gene. So I wonder if I could ask Dr. Blackstone to comment on where these are and what we're trying to learn with them.
Sure, Joe. So as you mentioned, Drosophila has several advantages as a model system. First is it's in vivo. It's an actual organism. It's not just a cell line. But one of the real great advantages, as you outlined, is that they reproduce so quickly, and the genetic studies can go very fast. So the advantage of Drosophila is speed. We can get answers quickly. We can do phenotypic analysis, like whether they're walking is affected, whether there's lots of phenotypes that we can see in a small fly that we can't see in a cell. So there's a big advantage there. Also, as you outlined, there's been a lot of previous genetic studies and a lot of biology done. So it's pretty easy for us to see patterns of abnormality, patterns of improvement that might give us more insight into how this protein is functioning. Another advantage is that in some cases, like in SPG3A, there's just one elastin protein. In humans, there's 3, and sometimes that can make it hard to understand the effects of one of them if there's 2 others that do the same thing. In flies, there's just one, so perhaps we get a more prominent phenotype that's easier to assess in this system. So, it's not like we only would do fruit flies. We want to do multiple different systems, and they each have their own sort of advantages and disadvantages. And in the case of this, I think we're going to have the opportunity to really clearly see the effect of removing what we'll call the bad elastin, the one with the mutation. And to just sort of clarify the types of mutations that we're dealing with these SPG3A and 4, in particular these severe forms, are there what are called missense mutations. There's just a change of an amino acid. So we think the protein is still there. So the question is it doing something bad? Would it be better not to have it at all? And that's, of course, what we can do with these experiments where we're trying to selectively deplete that using sort of not gene therapy, but genetic-based approaches. So a lot of the studies that we mentioned already for the cells, we're doing the typical drug approach. We're adding a compound and seeing if it gets better. The flies, and to some degree the mouse studies that we'll discuss later, allow us to also look at genetic approaches, and it widens the sort of range of different therapies that we're testing, again, with the hope that one of them will be particularly effective for these children.
Excellent. Thank you. if we move on to the next slide. This is the mouse model slide that we have alluded to. So a lot of the first 2 years of our experimentation, especially with the iPSC and with the Drosophila, has been to try and get enough information and maybe validation of previous hypotheses to then go ahead and work and test in the mutant mouse model. This is a mouse model that we have been developing with Jackson Laboratories, which is a world premier provider of mutated mice that is used in lots of research in biomedicine. And we're very pleased to be able to have them as working with us. And what we have done with them is to take some genetic instructions, which Dr. David Liu at the Broad Institute, as a world famous geneticist, has provided for us to be able to edit into a mouse model so that the mice have the exact defect of SBG3A and could be done with SBG4. So with this, our purpose is to see if we can grow generations of mice that actually have the characteristics of the neurologic defect that our children have, and if that's called showing a phenotype. If in fact they can show the phenotype, then we can go ahead and start to test out these hypotheses that we've been talking about. And I'm going to have Craig kind of carry on from here. But the first step is, do we have mice that actually show some kind of a what's called a phenotype, or the neurologic defect? Craig?
Sure, thanks, Joe. So one of the exciting things, and I know this is preliminary data about this new mutant mouse, is that it does appear to have a fairly strong phenotype. And not only is it strong, it's stronger than the other typical mutations that we see in SPG3A. And if you remember, that's exactly how it is in people. You know, there's some of these children with specific mutations are having a more severe form. What's, I think, particularly encouraging from a therapeutic point of view is that the mice seem to be showing the same type of pattern, that the specific mutation that we're targeting here is more severe than the others. And that, I think, has a lot of advantages. First is it will allow the mice to develop a phenotype earlier and more severe. It will be easier for us to do studies to see if we're improving that quickly. You know, we don't want to have to wait years to see if we have an improvement, or to have a very small improvement that takes a long time. One of the advantages of this mouse that I think you didn't discuss as much was that we actually put a human portion of the gene into the mouse. And that allows us to test for the gene editing, which is to go in and just fix the mutation at the genetic level. It allows us to fix it on the human piece of that gene. So it's the same kind of approach could be used in a person. So the mouse in some ways is what we call humanized. It has some it's not part human, but it has some human DNA that allows us to use the exact type of approach that we would do in a person in terms of fixing this. Now, not only will this be good for several types of gene-based approaches, this will also be a very good model for testing the compounds and the drugs that we'll be also investigating in some of those cell line studies. And hopefully those cell line studies will give us the type of information that allows us to optimize the design of our therapeutic trials in these mice. But I would certainly emphasize that having a strong mouse model for a disease, a neurodegenerative disease, is very, very valuable in terms of therapeutics development. And it certainly looks like from the initial studies that we have, they're still preliminary, but they look very, very promising.
Yes, and that's wonderful news. The other thing about mice, though, is that they aren't as fast as fruit flies in generating generations of them. So it takes several months to grow, to develop a cohort, and then several more months to test whatever therapeutic agent we're trying to test. So although the mouse model is much more definitive for us, it also takes much longer. And we're just now getting to the point where we think this later this spring, we'll have a large enough cohort that we can start in a staged way, looking through the 4 hypotheses that, once again, largely generated by the work that Dr. Blackstone had done at NIH and with other collaborators around the world. And as you might expect, we need to do these kind of in a staged way. We are planning to start with the lipid modulators and the BMP signaling inhibition, the first couple of things we talked about. Then move on to the more gene type therapies with the with so-called ASO therapy, and then finally the most sophisticated, which is the base editing or a kind of gene editing. I think to set expectations, we should recognize that to do all of this is going to take probably 1 to 2 years, but we'll be learning all along the way, including probably definitive learning these things kind of in order as we go. I don't know, Dr. Blackstone, if you want to speak to some of these hypotheses a little more, or anything else about the mouse model.
Well, I can emphasize that certainly we know a lot about where these proteins are in cells. And from our basic science studies, again, over time in different systems, we've identified either just studying the basic functions of the proteins or in some simple models like in cells and so on, have gotten suggestions that these pathways are effective in terms of at least ameliorating phenotypes that we see in cells. And again, that's not the same necessarily as the phenotypes that you'll see in a person or mouse or a fly, but it's a very good start. So again, I think that a lot of basic science over a period of at least a decade went into sort of identifying some of these promising pathways. And also, and I think you mentioned this, because these are what are called dominant disorders, where there's still one normal gene, the idea of removing the bad gene is always going to be attractive. Or of course, the best thing of all, if you can imagine, is to just go in and fix the genetic defect. And that's sort of that base editing. So I think that what is good about this is there are lots of different approaches to sort of the same problem. And I think that that's what we like in therapeutics development, because as you know, Joe, not everything's going to work, even if it's a great idea. And we have to have sort of several irons in the fire to ensure that we increase our likelihood of finding one that will be effective, or there's a possibility that combinations will be particularly effective, as have been shown for many other disorders. So again, I think there's a lot of strong biology supporting the approaches that we're taking, both the drug therapies as well as the sort of more genomic or gene-based related therapies. And as you said, that mice take a longer time to do these studies. But what's particularly encouraging about this mouse model is the mice are developing the phenotype very early in adulthood, which should allow us to speed that process up over typical mouse studies, because they develop the phenotype so quickly and because they worsen so quickly. That should allow us to assess the efficacy of our therapies much faster than you would typically see in most mouse studies. Very good.
Next slide, please. So in summary, I think we could say that the first phase of the programs that the Carter Foundation has been sponsoring, focused in the pluripotential stem cell and in the Drosophila, have really led us to be able now, with some better confidence and clarity, to go on to the mouse model phase, which is now getting started and will probably take 1 to 2 years. But I think we benefited greatly from all that we've learned for the last two, and I think it's increased our chances of success, hopefully successful therapy, and certainly a lot of clarity over the next 2 years. So I want to thank everyone for your attention today. I also want to thank our hosts, Ache, Feliz, and Juan Ruiz, who have sponsored the SPG3A website, and also have been helping us to give these updates to you through this website. If you have further comments and questions, please about the activities of the Carter Foundation, feel free to send them to me at avalon@comcast.net. So with that, I would thank you again, Craig and Ache, and say goodbye to everyone with the idea that we'll be back for another update in about 6 months.
Thank you.
Thank you.